Que sepáis que está grabado todo.
Bueno, ya nos has escuchado.
Okay.
Venga, vale, adiós.
Okay. Welcome to the second and maybe the last session in some time of this bioinformatics course. And in this session, we will talk about the clustering versus denoising processing of sequencing reads for metabarcoding analysis. And we will talk and see some reference databases. And the workshop we won't do today, we'll do probably next week in Petit Comité. Okay, so a little recap from last session. So we all start in the same base ground. We talked about what metabarcoding was. I want to point out again, it's just another technique to try to understand how the ecosystem functions. And it's just as valid as any other technique, as any enzymatic activity. It's just another kind of protocol, a bit more complex, to try to understand what goes on, in our case, in the soil and in the plants. So the principal objective of metabarcoding is the identification of the taxa that make up a community. It is mainly applied to microbial communities, but it is also can be applied to other animals and stuff like that. The typical workflow is you get a sample, you extract DNA, hopefully you get some DNA, you send it to sequencing, most likely still it's going to be sequenced with Illumina sequencing technology, and you obtain these FASTQ reads, which have a header, the DNA sequence, and then the associated quality of each nucleotide. This quality will tell you the probability that this is in fact a cytosine, and this is in fact another cytosine, and this is in fact a guanine, or it tells you, okay, we think it's guanine, but maybe it's a mistake, okay? So this is the basic first information you have from the sequencing platform. And what we do is bioinformatics with this to get biological information. The advantages are to identify microbial communities, which are hard to identify by analyzing in the lab by culturing, because you filter out unculturable bacteria or fungi. So we can also detect cryptic taxa, unculturable taxa, unknown species. This generates a lot of information to get a more or less complete picture of our community. There are some limitations to keep in mind always. There's this term called microbial dark matter, which refers to unknown taxa. Okay, we sequence, so for example, we get a sequence, we know this other guy in Australia, for example, got also these sequences, but we don't know what it is. And we also have to keep in mind that there's a barcode bias, which some species, some microbial species will not be amplified through PCR, and we will not detect it with this technology. What we did last time was clean the FASTQ reads based on the quality associated to each nucleotide. We removed the primers, typically here in the beginning of the sequence, and we filtered out low-quality nucleotides. So we obtained good quality reads to start working. What we're gonna do in this second session is, now we have clean reads, we'll try to understand how do we pass from a million of these, like a million sequences. It is a lot. How do we go from this to a community matrix? This is the starting point in any ecological study, environmental study. You have species, for example, alpha protobacteria, phenobacteria, gamma protobacteria. You have study sites, okay? Like this is, for example, Algaida 1, Algaida 2. It is the same as if, Pascalis goes to Algaida, he goes and he counts species. He obtains also this. With this, we can do all the ecology and downstream analysis and see what is happening in the ecosystem and in the community. But for now, we have this, which is really this, and we see, okay, how do we get from this to taxa associated with how many of these taxas do we have in the million reads? Because we might have repeated sequences like these ones, and we see how many times does this sequence happen. Okay, so it happens, for example, very abundantly. It might be a dominant species in the community. So that's the big question. How do we tell that this one is in fact alpha proteobacteria? This is the main objective of understanding how we can do it for today. So, what is evolutionary genetic divergence? Why does metabarcoding work? Why do we have biodiversity? It is because there's variation in DNA in all living things, and this variation makes life happen and biodiversity happen. It makes DNA diverge from a common ancestor sequence, for example, through evolution, time, errors, chaos, two sequences appear. And then these two sequences, which are at the core of maybe two different species, get separated into, okay, this is species one and this is species two. So thanks to errors in the polymerase, but also other types of mechanisms that provoke this variation in DNA, we get biodiversity. So this is great. Thanks to that, there's diversity in life, and life can adapt, and all the cool stuff that it does. It is also why we look for those high variables, hypervariable regions in barcodes, which will tell us, okay, this sequence is different from these sequences, because in this region, we see there are differences between species. The problem is the same molecular machine that enables biodiversity and life is also the one we use for sequencing. So this is kind of the big question here is, how do we tell apart that the polymerase, the variation in a nucleotide from G to T was originally in the sample, or it happened during the sequencing technology in which there are many, many, many PCR cycles? How do we tell this apart? So this is the big questions. How do you tell a real biological variant? Okay, you have two sequences that differ from one another. Sequence one, sequence two, they differ in just one nucleotide. But how do you know it's a real biological variant or it's an artifactual, fake, technique produced sequence? So this is the big question, and this is what leads to two different approaches, which in the end we'll see it's not that different. Clustering into OTOs or denoising into ASVs. Okay, so when this first problem appeared, when the first metabarcoding data sets were played, we, like researchers, had this question in mind. Okay, how do we tell, like, these are sequences, imagine these are sequences, and these ones, for example, are very similar, and you say, okay, how is this sequence real? How do I know that this sequence is real or a PCR product? They said, we're gonna cluster them together because we know that the biologically real sequences will always be more abundant than its derivated artifactual sequences. Biological real sequences will have bigger dots, which represent abundance. And from these sequences, during the sequencing technology, some sequences get formed due to PCR errors. So the assumption is the real sequences will always be more abundant than their derived artifactual PCR product sequences. And by clustering them together, we lower the effect of fake sequences. Okay? And normally, lately, the identity threshold to cluster sequences together is 97%, sometimes 98 or 99%. So I'm going to ask you a question to see if you got clustering, the main idea of clustering right. Imagine these are all similar sequences that are 100 base pair longs, 100 nucleotides. If we cluster at 97% identity, what is the maximum number of differences between all of these sequences within this red circle? If we keep in mind, these are 100 long base pair long nucleotides, and we cluster them at 97% identity. So only 3 nucleotides will differ from all of these sequences, and we cluster them together.
But I have a question. You
Sí.
always set the identity threshold in 97.
It depends. We'll see later.
Nine.
It plays a big role. So there are multiple ways of clustering. What I showed earlier, ¿tenés calor?
No.
¿Está bien? The idea of that I showed in the previous slide is that we start this from the data we get. So it's de novo. We got all these sequences, and within our data set, we try to put them together. This is computationally very difficult, and there are also multiple ways of addressing this, and it's not easy. At the beginning in 2016 or even earlier, this was how first OTUs were formed, but it is computationally intensive. Nowadays, we can do it with supercomputers if we know how to use them, which could be a nice option. Here I cite an algorithm that does de novo clustering. What typically happens is closed reference clustering or open reference clustering. So what accelerates a lot of the clustering is to align our data set with the database's sequences. There are databases out there that store many sequences, and many of those sequences are even assigned taxonomically. So to speed up the process of doing this, we first align our sequences from our study to reference databases. And if the alignment of, for example, these sequences, this one sequence, the first one, aligns with one in the database, this one is clustered to a reference database OTU. Okay? Unaligned sequences that do not match the reference database get discarded, eliminated, which is a problem. It's not a problem if you're studying our gut microbiome nowadays, because the database of gut microbiota is complete. Like, there's a lot of research, lots of years, lots of studies, lots of data on our microbiota. But what if you go to Antarctica or you go to somewhere else? Environmental samples are less studied and are more diverse too. So there are going to be more rare taxa that will not align to the reference databases and will get eliminated. So we lose information about the community. So that's why combining these two methods appears open reference clustering. What we do is the same as this, okay? But instead of eliminating sequences that do not successfully align to databases, what we do is do open reference clustering with those unaligned sequences, with those sequences that we couldn't align. So by combining these two, it's a bit of the best of both worlds. I would use open reference clustering for generating OTUs. Another thing that most pipelines of clustering produce is a consensus sequence, which can be thought as the centroid or the average, the mean of all the sequences. So imagine a cluster of 3 reads, read 1, read 2, read 3. They are similar within the 97% threshold, okay? They differ in few bases, but they still differ in some nucleotides. What a lot of pipelines do is build a consensus sequence that represents that OTU. Okay? What I want to ask you, is this consensus sequence real?
Probably not.
It is a consensus sequences. It will depend on your original data. And if someone else does another study or even resequences your same sample, he will get different reads, and his consensus sequence representing the OTU will be different. So this is a bit of a downside of OTUs. This is why denoising appeared. So what is denoising? Who can tell me what is written here? Denoising what? Reads. Okay, so the idea is to remove the noise. In any signal, in information theory, you always have noise. We have our sample, we sequence it, and we get a signal which is fastly reads. But every signal has some noise. We have our sample, we sequence it, this is what we get, which is a signal that is noisy, and denoisers will try to remove that noise, that's why they're called denoise, and get a clearer signal of the community, the biological information that is in the sample. By clustering into OTUs, this noise gets incorporated into the information, which is at the baseline of the rest of the study. Denoisers try to eliminate this noise and get a clearer picture that has more fidelity with the original composition of the sample. There are a few denoisers. There's data2, the blur, there's also unoise, but all of them try to do this: remove PCR artifactual sequences. And how they do this is with statistics, models, probability. Um, so I'm gonna again put in front of you an example. This is our data set. We have 3 sequences, and we have how many times these sequences happen in the data set. So there are, for sequence 1, 3,503 counts. This means sequence 1 happens 3,503 times in all the data set. We have sequence 2, which is a bit less abundant, half abundant, but it differs from sequence 1. This is why it's different. It's called sequence 2 because it differs from one nucleotide. This is a nucleotide transition happening here. And we also have sequence 3, which is really low abundant sequence, which has, when compared to sequence 1, has one nucleotide transition. Denoisers take into account sequence counts, how many times the sequence happens in the data set, and also take into account, not shown here, the inherent quality of each of these nucleotides. By taking this into account and modeling how errors of PCR happen and errors in sequencing happen, they can tell or infer that this sequence is probably, most probably with a p-value of super low p-value, that this sequence is a PCR product of the technique. This sequence did not exist in the original sample. This sequence was created from these sequences during sequencing, during the technology, during the obtaining the signal. However, because of sequence 2 only differs in one base, so it's really close to sequence 1, but it's so abundant that it most probably is this green dot here, okay? And we consider it a real variant. This is because PCR errors during sequencing are random. They can happen at any time. So a polymerase makes an error, and it can happen anytime. Real biological evolutionary variation in DNA is not random. And it is what tells us that one species is one, on one other species is another, based on DNA barcodes or DNA sequences. So I would ask you, what would you choose if I gave you now a data set? What would you choose? Clustering into OTUs, or would you denoise into ASVs?
Is the same the taxonomical resolution in both of them?
No, the taxonomical resolution, well, it depends. If you cluster OTUs at 99% or 100% similarity, it's more or less the same as ASVs. But denoisers typically take into account the quality of the bases and how abundant. So typically, OTUs will be have less taxonomic resolution than ASVs. So Marco, what would you choose?
Depends on the objective.
Okay.
Because if you want to describe, let's say, new species or have the maximum diversity, you will do denoising. While if you want to, let's say, do a functional approach, maybe identifying families, you don't care about two sequences are like two families are two reads are a bit different. Even if it's on a high count, you can put them together.
I would do that. Like before working all this year in this, I was really into ASVs, and because I got into how the denoising happened. I was like, well, this is amazing. We can reduce the noise, get a clearer signal. But then the taxonomic resolution is so high. It is sequences. Do you really need taxonomic resolution? That high resolution. So in the end, if I would continue to analyze microbial communities, I would go with that mindset. That it depends, and we will see. I think now, if I had to do all of this again, I would combine both approaches, and we'll see why now. And this is really interesting, I think. So here are some differences, resumed differences between OTUs and ASVs. Some advantages of OTUs is reference-based clustering can work really well for well-studied and well-mapped environments like the human gut microbiota. It is fast to do and easy, more or less, to do with the reference. And it also approximates to a concept of species. And it represents a solid basal taxonomic unit. We'll see this a bit later. The disadvantages, I would consider consensus sequences. Are they real biological sequences? Can they be compared to other studies? ASVs can be compared to other studies because we assume that the denoising method will get real biological variants. So we can compare single ASVs with other studies of ASVs, and they offer a higher taxonomic resolution. And the question is, do you need it? Some disadvantages of OTUs, apart from consensus sequences and that they're not comparable, is the incorporation of errors in the errors of polymerase that happen during sequencing. Some disadvantages of ASV: they're a bit more computationally demanding if you have many reads, and it's also like the higher taxonomic resolution. Is it too much resolution? Do you really need it? So why do we cluster OTUs at 97%? At the beginning, at like 2010 or even before, OTUs were clustered at 95%. Okay? Then they said, okay, we have to cluster them at 97% for 16S. Huh? Keep in mind 16S barcode, really important. And then this paper came out, and the identity threshold keeps updating. It depends on at what level you want to cluster. And here is a bit like, okay, we want to have a starting point for downstream analysis where we have species and their composition. Just like what I said before, that Pascalis goes to Algaida, sees in the spring, he looks at the plants and he sees, okay, this plant, there are 100 plant A, okay? He knows that this plant is a species. He doesn't care if there's variation between the species because he identifies them morphologically, more typical of classical taxonomy. However, here we're working with molecular kind of taxonomy or phylogeny. So, what is a species? What is a bacterial species? In this paper, this guy, Robert, said it depends on the length of the barcode. Depending on the length of the barcode, for example, 16S, if it's full length barcode, at 97% identity, you already see differences in the probability of it being different species. Okay, so how we understand this graph is that if we cluster at 96% similarity, might be a too harsh of a threshold because we will cluster and clump together OTUs into same species when they're actually probably not the same species. And we'll see that with full length, we can cluster at 97, more than 97% identity. However, for shorter regions, like the hypervariable V4 region, he recommends clustering at 99% identity. Why is it? A shorter region will also have less potential of telling you this species is different from this other because there's less nucleotides. So there are going to be less differences, and the longer your barcode is, the more easily and more confidently you can tell, okay, this species is different from one another. Because in a V4, for example, barcode, you might have 10 mutation or transitions or nucleotide differences. In a full length of the same species comparison, instead of 10, you might have 35 differences. So the more differences you get, the easier it is to tell you, it is for you to tell that two species are different. This, I looked at it a bit yesterday because what is a microbial species? What is a fungal species? What is a bacterial species? Like this dives deep into another complex subject of species delineation and identification. I mean, we are aquí in Opistogonta, like within this, like we are just very small, like in the tree of life diversity. And we're trying to study all of this with a V3, V4 region. So we have to understand the limitations also of the metabarcoding technology. We cannot assess the species resolution. All the experts nowadays tell you, unless you sequence like the full length 16S operon, or you do multiple lossy sequencing, meaning you sequence of the same sample, the 16S, the 18S, and some other gene, you will not get species level resolution for bacteria because all of these are bacteria. Like they're extremely diverse, and we often think we have this bias, which is natural, of thinking of species like animal species, which classic evolution, classic biology, and ecology works in that realm. We're trying to apply and use the tools we have developed through a century or many years of study of ecology to microbes. But it's a bit different. What is a microbial species? This really shocked me. So the criterion of species delineation, 97% similarity of the 16S region, if we kind of apply it to animals, all the primates would be the same species. Would be the same species as chimpanzee, as orangutan. We would be the same species. So it's it's a bit complex. So just so you have that in your mind, comercial Google. After the after denoisers appeared, like this paper came out and an opinion formed that exact sequence variants should replace OTUs in microgene data analysis or metabarcoding. So this is a pretty big statement. Of course, this is also clickbait because within the article it says, okay, this is developed for 16S barcode. You might want to change the parameters of the algorithms or other barcodes, but this is like a powerful statement. And does it really matter then if we cluster two OTUs or ASVs? There are several papers, and there are more, that see an effect in the biological signal, like alpha diversity. Depending from the same data set, depending if you cluster or denoise, you get differences. Like, so it is important to take into account this, and it's also a bit complicated to like, there's no consensus of a pipeline, you should do this. Depending on what you use, you will get different results, okay? So it's not like in the lab where there's one or two protocols and you kind of trust the protocol, right? Juanfra, tú cuando haces un protocolo, confías en la señal que te da eso. Bastante, la mayoría de veces. So it's like, do we take OTUs or ASV? It still depends, and we'll see later. For fungi, which is mainly by sequencing ITS, many experts studying fungi and doing metabarcoding fungi reject the ASV or the denoising. No, they reject, careful because it's not the same. They reject the use of ASVs as the basic taxonomic unit for your study, because they say ASVs do not approach the species concept in fungi. Okay, the original data 2 paper acknowledges that if we use data 2 on fungi, some values of the algorithm should be changed because it was developed for 16S. ITS is these regions here. So small SU, small subunit of the ribosome RNA is in prokarya and bacteria would be 16S, and in eukarya we use 18S. These regions are more variable than 16S. So the ITS regions is way more variable. So it has higher intraspecific variability. It has high intragenomic variability and high intra-individual variability in heterokaryotic fungi. What does this mean? Intraspecific variability means we have this construct of, okay, this one mushroom is a species, but then when we sequence it, we see there are variations within the same species. This would be intraspecific variability. There's also intragenomic variability, which within the same like genomes, there's also variability, and there's also high intra-individual variability, meaning some fungi have multiple nucleus, multiple copies of their genome, and there's also variability in that. And as well, this operon is repeated in the same genome. So this is what the intragenomic variability refers to. So fungi people kind of reject the idea of ASVs or exact sequence variants as the baseline taxonomic unit that proxy as a species. Remember, we want to get the community matrix similar to what botanists or naturalists use for study ecology. So they say, okay, they say, for example, in this paper, you can use denoising, but then you have to cluster because ASVs will not represent equivalent to fungal species. They represent higher taxonomic resolution than what we need with the use of ITS. Also, ITS, some people use small subunit barcode, for example, for arbuscular mycorrhizal fungi. So there's there's no perfect barcode. This we saw in the last session. So you're always limited by the barcode you use, and some barcodes will work good for one branch, but will not work good for all the other branches. And even within here, which where it's where eukaryotic fungi are. For protists, I don't know much about. Hina knows for sure much more.
For my proteins, ITS sucks.
See, exactly. So, there's too much variability, I assume, right? So you use 18S, right?
And even SSUs too.
Okay, the second,
Sometimes, for
some
some
other
reason.
subunits, yeah. So it depends on what branch of life you're studying. So it's really important to know your barcode. This is the main conclusion for me is really understand your barcode and really know what genomic region, what molecular region you're studying and how it varies within your field of study. So this is really important. And of course, I arrived here last year. I knew basically nothing about bacteria and fungi, and this is a conclusion I've reached now after a year of going a bit blind and thinking I was doing it right, but no. This paper combines denoising or de novo clustering, and then does a second level grouping. So they see denoising or de novo clustering as a first grouping, okay, of sequences, and then they do a second grouping to approach the concept of species in their field of work, which are protists. So I read in depth this paper, which does metabarcoding in animals, but I thought their approach was amazing. Not only I will not go into this deep, but they adapted the denoising to the denoising algorithm. They adapted it to the COI gene marker. COI gene marker is coding. What we saw before, sorry, IDS, it's not coding. It's ribosomal RNA. IDS is not even ribosomal RNA. 18S, 16S are ribosomal RNA. Their structure to which proteins attach and form the ribosome, but they're not coding like COI. So there's intrinsic variability um, to codons. So codons are these 3 nucleotide long sequences that tell you, okay, this is this one amino acid. The ribosome reads it, and it gets one amino acid, and they stick amino acids together to form proteins. Do you remember from biology class the big table of codons, where there's ACC, ACG, AC0, whatever, code for leucina, okay? Lots of times, depending on what position in the codon, 1, 2, or 3, there's going to be higher or lower variability, intrinsic, like in any coding genomic region. In any gene, the position in the codon will tell you if it's more or less variable. So they adapted the denoising to that. So they took the unoise algorithm and they adapted it for their barcode. So they really know their barcode. Like they understand the molecular entropy of their barcode. And what they did was denoise first into exact sequence variants, and then they said cluster. And they took what, like the idea Marco said before. They said, if we want to study the most basic ecological indices or how the community is formed, we will use OTUs because they approximate our understanding or our concept of species. This is easier because it was for fish. So they already know, okay, the percentage of identity in this barcode for different species. So they knew at what percentage to cluster to approach the idea of a species in the fish's realm. Okay? So they used OTUs for alpha, beta diversity, biogeography, ecological indices, like the typical ecological study. But then they said, okay, we can use exact sequence variants in combination with OTU to see metaphylogeography. What is phylogeography? Means within the same species, how does evolution happen? So you have some species here of fish in Cádiz. And you also take some, there's this migration route, and there's different migration routes, for example, or path, and you say, okay, I'm gonna sequence the different regions, the same species, the different regions. And you can see, they saw that this could be used to understand how evolution works. Because imagine you sequence a wide community of a species in one point, and then you study this distant, like isolated niche of the same species, and you maybe see, okay, we consider them the same species, but we are already seeing evolution at work in some molecular divergences. So this inspired me to go deeper into the subject. It's a bit of this last part of the presentation was about. And yeah, so I would say combine both approaches depending on what you want to achieve. Uh, Referring databases, there's this barcode of life database, which has animals, plants, fungi, and protists. I would use this one for animals. I don't know for plants. For bacteria, I would use Silva mainly. I want to try this one.
This is my boss. I mean, my master supervisor, Antonio. This is what
Sí.
we're trying to do. Yeah.
Okay, so yeah, he's also working with 60ness.
Yeah.
So he curated a database, because what happens with databases is lots of times you go to NCBI, it's just people like throwing sequences and no one is like organizing, assigning, like it's just a bunch of sequences. So some databases will be more curated. Okay, they will have low redundancy.
Yeah.
For fungi, I've always used Unite. There seems to be a consensus that Unite is the way to go for fungi. There's also these databases with fungi that has geographical information associated with it. Also differences with arbuscular mycorrhizal, because if you want to know about arbuscular mycorrhizal fungi, normally you use not ITS but 18S. And for protists, which I don't know nothing about, there's this database. I haven't really explored protists a lot. A last slide, and we leave because we have to leave very fast. There are aligners and there are classifiers. They're not the same. Aligners will take a sequence and align it to other sequences and will give you this result. Okay, you say, okay, these sequences I put is this one virus. But sometimes you have like here 100% identity, and sometimes you put a sequences and all of these are 100% identity, and you see, okay, which one of these are my sequences? So classifiers give you like one result. This is the main difference. And classifiers are hard to understand. And we will not go into them because I cannot really explain them yet. Yeah. So I think this is it for the session. It goes into species concept and the baseline of like the basic stuff from which to then study. Like how we get the enzymatic activity, like nanomoles por hora por gramo de sol. So we want to get a clear understanding of what our unit is. ¿Qué unidad usamos? ¿Y por qué la usamos? Y para eso, pues, tú descubres mucho.
Pero en verdad, si quieres datos masivos, yo creo que es tan ahí a especie.
Sí, yo
Más arriba, ¿no?
estaba usando ASV. Claro.
Pero en comunidad un poco más grueso, ¿no?
Si ahora tuviese que hacer, por ejemplo, para bacterias, usaría ASVs y los agruparía en otros. Eso haría. Para comerme.
Claro.
Y sí que tendría mucho en cuenta las diferentes barcodes. Como el ITS. Yo estaba tratando, hacía el pipeline para 16 y hacía copiar, pegar, venga, lo mismo para ITS.
No vale.
Eso, muy mal.
No vale.
¿Sabes? No me...
Claro, pero, o sea, a mí te llegan datos los que te han llegado de 16 eso.
ITS y también tenemos de 18 eso.
De las mismas muestras tienen las tres cosas.
Pero bueno, es complicado y hay muchos puntos en los que piensas, guau, aquí se me puede sesgar muchísimo la señal. Pero bueno, aún así es súper útil, ¿eh? Y se han sacado un montón de resultados respecto a esto.
Vale, nos tienes que enseñar con la parte de datos y esa movida.
Sí, la semana que viene nos
yo
ponemos
no
ahí
quiero, pero
con Clara también y con Marco también, que también lo tiene que hacer.